Hi, my name is Tyler DePrano, and I will be doing a presentation on the alternative pathway of the complement system. So, what is the point of the alternative pathway? Alright, so we know that through the classical pathway that we can have antigen-antibody interaction, and that antigen can then be identified to the complement, and a membrane attack complex can form, right? So this can happen, and the membrane attack complex will eliminate any antigens based on whether or not any bound antibodies are there, or in the case of the mannose binding lectin pathway, if there is any sugars present on this antigen, we can also form a pathway through that, through a complement, and a membrane attack complex will form. So in the alternative pathway, the real defining point here is that we aren't going to be using either of those. So this is all interrelated in that the membrane attack complex can still form through all three pathways. But the difference is how the pathway will start, right? So, what's the real difference? Well, complement can kill things that the rest of the immune system can't, okay? But how? How is this alternative pathway identifying the rest of these antigens? Okay, if it's not going to be with mannose binding lectins, if it's not going to be with antibodies, what is actually going on? Well, the key point of the alternative pathway is the complement protein 3, or C3. Okay, so C3b inhibitors are present on host cells. Okay, so C3b inhibitors are not going to be present on these antigen cells, right, or antigen surfaces. And that's going to allow the complement to attach. Okay, so if C3b isn't degraded, it will attach. It will attach to these surfaces. But on host cells have proteins that will break down the C3b protein. Okay, so if C3b protein can bind to the surface, it can start this pathway. Okay, also, C3b will bind preferentially to the antigen surfaces, right? So this is basically just what I've been going over. Okay, it's going to bind preferentially because these surfaces are going to be, they're going to be charged. So it's going to bind preferentially to the surfaces, and it's not going to be broken down. Okay, so those are the main 2 points that make the alternative pathway work, right? So how is this an alternative? That's the real question. Well, it doesn't require any of these antibody-antigen interactions, right? So that's what I've been going over. You're not going to need any antibody to bind. It's also not going to require any sugars, okay? So if it doesn't require any of the sugars that are recognized, so mannose is a sugar. Our bodies, as far as I'm aware, use mannose, but very, very rarely. Okay? But there's also other carbohydrate patterns that these MBLs, mannose binding lectins, can recognize. And that also can include stuff like the NAG sugars present in peptidoglycan. So peptidoglycan is going to be around bacteria, and MBLs can recognize it, and they will go and attach, right? But the alternative system doesn't require that. It just requires that the C3 be able to bind. So C3b be able to bind. Okay? So let's go and take a look at what is actually happening with this alternative pathway. Alright. So you can see here C3 right here is going to be degraded spontaneously. So it's happening. This is happening all the time, right? So C3 is always being degraded, and it is being formed into C3b and C3a. Okay, and if you didn't know, C3a is an aflatoxin, so it's going to help with inflammation and such to deal with any infection. But C3b is what we're interested in here for the alternative pathway. C3b, as you can see, will go down to the cell surface, and it will bind to the cell surface. Let me just see here if I can get, there we go. So C3b is binding to the cell surface, and what is happening after that? What is this FB that we're seeing? Okay, so this FB right here is called factor B. Okay, so factor B, this will bind C3b when it is binding to the cell, okay? And you have to recognize that also that there's going to be excess C3b. So if it's not being degraded normally and it's sticking to the cell, what happens is when it binds the cell, the proteins that degrade it aren't going to degrade it anymore once it's bound, okay? So if you picture you have an antigen, so you have like a bacterial surface, right? And you have a host surface. C3b is going to bind the bacterial surface, and these proteins are going to be like, whoa, this guy is in our house, we have to kick him out, or we have to destroy him. So they're going to go and start cleaving C3b on a host surface. Now on a bacterial surface, C3b is going to bind, but the bacteria don't have those proteins that can recognize that C3b needs to be destroyed, okay? So C3b is going to stay there. And then you're going to eventually build up some excess C3b, and, factor B is going to come along and say, hey, I like this guy, and he's going to stick to him and bind. Now, once factor B is bound to C3b, factor D is going to come in, and it is going to cleave factor B, okay? So now we've got two factors, factor B and factor D. Factor D just cleaves factor B. Okay, so you've got a BB fragment and a BA fragment. So it's just like C3A and C3B, but with a B fragment of B and an A fragment of B. So C3B, BB will then be what's left. The A fragment of factor B is going to just go off, and we aren't going to focus on that anymore. So C3b, BB, then is also going to act as a C3 convertase. So what that means is it's going to cleave C3 protein into C3A and C3b. And you're going to get more C3b built up on the cell surface because, as this C3 convertase here is cleaving C3, the C3 that it's cleaving is going to be near the surface. So the C3b that's been cleaved will be like, oh, the surface, I want this surface. I want to eat what's on this surface. I want to bind to it. I like the surface because it's already near it. So it's not going to bind to anything else or be degraded. It's going to bind straight onto the surface. And what's going to happen is you're going to have a positive feedback loop in that it's creating more C3 and more C3b. And again, eventually factor B is going to come in, and factor D is going to start cleaving more, and it's going to just increase exponentially as it goes. So that's a positive feedback loop. And what's going to happen also is eventually you're going to have a C3b that will bind to the C3 convertase complex, okay? And that's going to form C3b, BB, C3b, okay? So it's a lot easier when you can see it like this. And that's going to be a C5 convertase, as you can clearly see. And what that is going to do is it's going to cleave C5 into C5a and C5b. C5b is then going to start your membrane attack complex, and from there that cell or antigen is going to be lysed and destroyed. So that's an overview. So just in case anyone's following along, let's just go and just have that laid out very simply instead of all the different shapes. Because while this is easy to see, it's not necessarily easy to remember without being able to see it. Okay. So we've got C3 protein. That's what we're going to start with. Alright, so we got C3. C3 is going to be spontaneously degraded because that just happens over the course. So we've got C3, and it's going to be spontaneously degraded into C3b and C3a. Okay? Now this C3b is going to bind to the cell surface, and factor B is going to bind to it. Okay. So once it's on the cell surface and factor B is bound to it, factor D cleaves factor B. And the B fragment of factor B is going to stay bound to C3b. And we'll be left with C3b, BB. Okay? So that's what we have here. From there, C3b, BB will act as the C3 convertase, and it's going to break down more C3, which is eventually, which is going to be broken. So it's going to break this down. It's going to break down C3 into C3b. So this C3b BB complex forms more of this. The C3b will then go and stick to the cell surface and form more of this, and we'll eventually have more C3 convertases. Also, the C3b is going to come back over here. So it's going to come over to C3b BB, forming C3b BB C3b. Which is going to convert C5 and start our membrane attack complex. So it's going to convert it to C5b. Alright, so how is this regulated? Because that's really what's important with the alternative pathway. If you don't have any regulation, then this is going to go and just kill host cells too. So you need to have stuff that can protect these host cells, right? So you have factor I, and what it does is it inactivates C3b. So factor I will just go and cleave C3b. That's what it does by inactivating. It's cleaving C3b. So just imagine factor I just clips right along the C3b protein. Okay, and factor H is actually involved in the removal of the BB protein. So if BB is bound to C3b, it's going to, factor H is just going to come along and say, nope, we're not having this, and just clipping that BB protein, getting rid of it. An interesting thing here is that factor H, you need to know this for why exactly these pathogenic cells are attacked and our host cells aren't. Factor H is required as a cofactor for factor I. So for factor I to work, for it to break down the C3b, factor H has to be present because it's a cofactor in the reaction. So factor I actually requires factor H, whereas factor H doesn't require factor I. That's important because factor H is what recognizes the pathogenic cells. Okay? So it recognizes substrates specific to host cells. That's what factor H does. Okay? So what kind of substrates are we talking about? We're talking about acids, like sialic acids, as well as GAGs, which are glycosyl amino glycoproteins, or I'm not too sure. They're carbohydrates. So the factor H is specific. It can recognize the things that are specific to host cells, and it's activated by that. So it'll start removing BB protein. Factor H will be activated and start removing the BB protein, and then it'll also allow factor I to start degrading the C3b when it recognizes these substrates. And that's really the very core of how exactly we are able to figure out why complement only, the alternative pathway only attacks pathogenic substances or antigenic substances. I should be using antigenic actually instead of pathogenic, because this will attack non-pathogens as well, as long as they are antigens.